Hello, my name is Carla Rubio Sandoval. I'm a PhD student at Bremen University, Germany, and I will present a research I'm developing with my collaborators entitled Mollusk Shells for AAR, dating the quaternary shorelands along the coastline of Camarones, Chubut, Providence, Argentina. During the quaternary, shorelands have collected information of sea level changes. The geological records related to the past sea level position are known as sea level indicators and allow us to reconstruct the sea level high stands during the interglacial periods, which are the Earth's warm climatic intervals. Knowing this information enables to develop projections of sea level of a future warmer world. Beach ridges are one of the most common sea level indicators along the coast of the southwestern Atlantic. A beach ridge is a wave-deposited ridge running parallel to a shoreline. It is commonly composed of sand as well as sediments worked from underlying beach materials. The beach ridge deposits of Camarones coastlines are typically composed of a sandy gravel matrix rich in pebbles and shells. However, these deposits lack material commonly used for the age constraint of coastal deposits. For example, another type of sediments for luminescence analysis or corals for the uranium-thorium method. To date, numerical ages have been determined by electrospin resonance and uranium-thorium analysis of shells of the marine mollusk Amegnomia antiqua, applications for which are some concerns. Why? Because mollusk shells are an uranium-thorium open system. This means that at the organism's death, shells allow the infiltration of water or other materials causing the incorporation or leaching of the uranium and thorium. As a result, the uranium-thorium levels are alterated. An alternative for establishing the geochronology of this deposit is the amino acid racemization method, or AAR. This method analyzes the extent of the protein degradation within shells and estimated time elapsed since the cessation of the protein formation, commonly equated with the organism's death. How? First, we need to know that there are two possible forms of the amino acids, levo and dextrohero, which are mirror images of each other, just like our hands. Roughly, all the proteins in living organisms consist exclusively of the L amino acid due to a chemical enzymatic selection. Following the cessation of the metabolic reaction due to the organism's death, levo amino acids are reversibly transformed into dextro until an equilibrium mixture of both is obtained in a process known as racemization. Therefore, racemization has been directly related to the time elapsed since the death of the organism. The AAR method used to date mollusk shells of quaternary marine and coastal deposits is well established, providing relative ages to a stratigraphic sequence in form of delta and L values. In this study, we use samples of Amegnomia antiqua to assess its suitability for AAR analysis in 6 Pleistocene field sites along the coastline of Camarones locality. You can see the areas in this map. These field sites represent beach ridges with maximum elevations ranging from 9 to 30 meters above high tide. As you can see in this figure, when we plot the delta Al values from the aspartic and glutamic amino acid, beyond the Holocene ridge, the Camarones beach ridges could be associated with at least 2 interglacial periods, consistent with previous published results, for example, the MIS-5 and the MIS-11, although some discrepancies are presented. The AAR results also show mixed age deposits. See the red triangles, indicating that reworking of older deposits into younger ones. Our initial results show that AAR analysis of Amegnomia antiqua is able to discern interglacial deposits of different ages and can be used to assess and complement electrospin resonance and uranium-thorium results. This project has received funding from the European Research Council under the European Union's Horizon 2020 Research and Innovation Programme. Grant Agreement number 182440. Thanks for your attention.